Salemi, welcome back to Device Talks Tuesdays. It's great to be here in the comfort of my office. We had Device Talks Boston last week. Wonderful to see people in person, but nothing like just a comfortable chair to sit back and watch a really interesting presentation. So we have one of those today. We brought back our friends from Nelson Labs. They were a big hit last year. We knew we wanted them to do another Device Talks Tuesday this year, and they were gracious enough to return. They have a new panel and a new topic, as you can see, current regulatory acceptance of alternative testing for biocompatibility. I said that without tripping over it, which is great. Before we get into this important discussion, remember this is available on demand. So if you're watching this now and it's occurring to you that a friend, a colleague of yours would benefit from this conversation, and I'm sure it will, please let them know they can watch it. They can just go to devicetalks.com. Can register, sign up, watch it live, watch it on demand, but it will feel as if it's live. And they even ask questions, and we'll make sure those questions get over to our friends at Nelson and our panelists today. The slides for this presentation, there's a great presentation for you. It is available in the resource widget, so make sure you walk home with those, or you're probably already home, but you know what I mean. Get the slides, take a look at them. And of course, I mentioned Q&A. We'll have Q&A during the presentation as well, or at the end of the presentation. So if you have a question, don't wait until the end to ask it. Please throw it up there in the Q&A field, and we'll know, therefore, how many questions we have to plan for, at least get a sense. So please just ask as they come to you, and be as specific as you can be so we know exactly what slide you're referring to. But no need to go crazy, of course. And finally, if you are using social media, use hashtag Device Talks Tuesdays. Real quick, we have our upcoming meeting, Device Talks West, October 19th and 20th in Santa Clara. You can go to west.devicetalks.com for more information, but don't go just yet. I'm working on the agenda for that. We have some keynotes up there, but give me a month and then go check it out. It'll be great. And of course, we have our Device Talks podcasts always, Device Talks Weekly, Stryker Talks, Medtronic Talks, and Tuna Talks. They're all available on devicetalks.com, available on various websites. And of course, all the podcast players you can think of are up there as well. So please do check those out. All right, now let's start the show. I'd like to thank our friends at Nelson Labs for putting this together. And I would like to hand over the control. Thor, are you kicking us off, or you're going to MC from here on in?
Well, I can kind of introduce my friends, and then I'll turn it over to Audrey for the presentation. So first off, I want to thank Kelly and Ron for joining us today. Good friends of mine, known them for a long time, and I can't think of a better group of individuals to kind of talk about these in vitro alternatives to animal testing and the current regulatory acceptance. I also want to thank Audrey, who I got to come and do a presentation for us on where the in vitro methods are. So as you can see, Ron is the current principal toxicologist at Risk Science Consortium. Sorry, Ron, I butchered that. And then also Kelly Coleman is a distinguished toxicologist in Medtronic. And like I said, we're both very happy to have him. I'm now going to turn the time over to Audrey to do a quick presentation. She's our senior biocompatibility expert here at Nelson Laboratories. So Audrey, turn the time over to you to fill us in with some in vitro alternative work.
Great. Thank you, Thor, and thanks everyone for participating today. So I just want to encourage everyone to put your questions in their Q&A. This is an excellent panel. It's great to have Ron and Kelly both on here. So take advantage of the expertise that we have available today, and please put your questions in. So the goal of my presentation is just to give a very brief overview of some of what we're currently doing, what's on the table for in vitro methods, so that we can spend most of the time discussing where we're at from regulatory perspective, address challenges, and also address your questions. So this will just be brief as I step into some of our more well-known approaches to alternatives to biocompatibility. So our first one here that I want to talk about is extractable unleashables and toxicological risk assessments. So using chemistry and toxicology, we can really address a lot of the endpoints that are identified in ISO 10993 Part 1. So you can see on this slide, I've just taken a snippet from table A1 from that standard to highlight the biological endpoints that need to be addressed based on, the type of patient contact that a device will have. For chemistry and toxicology, the most easily targeted endpoints are the systemic endpoints. And I've marked those here with some arrows. And you can see all of the systemic endpoints, chronic, acute, subacute, subchronic, and then also some other endpoints that we like to look at through chemistry: genotoxicity, carcinogenicity, reproductive and developmental toxicity. So just a brief thought of how we approach this is that we run the chemistry and identify compounds. In here, I used an easy one like caffeine, which all of us use on a daily basis to balance out our moods to some degree. And so we think about, it's the dose that makes the poison. And that's really what we're looking at from a toxicological standpoint. So when we get to a new normal, we're quite happy with our dose of caffeine, but you can see anything in abundance can cause, severe adverse effects. And so that's really what we're looking at when we do toxicology. We look at the chemistry data, identify those compounds, look at the quantities, and then go to publicly available literature to see what known values, known hazard values, or safety values that are identified there. And toxicologists will then apply that data back to the patient exposure of the medical device. So we really like this approach because you have a lot of data that you get access to. The quality is great because it can be very quantitative, very specific, targeted. You're not usually waiting on a biological system, right? You don't have to wait for cells to respond or animals to come in and be, get attributed or acclimated to their environment. And it's typically less expensive when we think about chemistry and toxicology, less expensive than some of these long-term animal tests. So I've got a big FDA stamp on here because we do have great approval from FDA, and we are seeing a lot of regulatory bodies actually asking for chemistry. So if some folks want to go a traditional route and go back to animal testing, it's more typically that they'll be requested to do some chemistry. So this is a great success story on using an alternative to animal testing. The next one, which has been a hot topic, I know for Kelly and I for a long time because we've collaborated on incorporating the in vitro irritation method into the standards. And this was added to ISO 10093-23, which we're very happy about. So the in vitro method here uses reconstructed human epidermis. And I've just put some images up here for informational purposes. You can see on the left where there's the tissue staining to show what it looks like live, like from a human. And then also what we can grow up in the lab is on the right. So what we care about is to show similarity between human, what we see in live and what we can see in the lab, and also that we're getting that great tissue and cell differentiation. So just to highlight this method and give everyone some perspective, I thought it'd be great to just show you some of the tissues and what they look like as we work with them in the lab. This assay is based off of cell viability. What we get is on the left, we'll get tissues coming straight from facilities like Matec or Skin Ethic. And so right now, these tissues have been acclimated. They just take a day or so to recover from shipping. And then we run through an exposure process where they're exposed to the extract for a couple of days, and then we remove that extract, and then we stain it with MTT. So many of you may know MTT as a cytotoxicity stain, and it's the environment that we apply the stain that makes the method, and that's really important to remember. So even though we use MTT for cytotoxicity testing, we're applying that stain in a more complex system. And what you can see here on these plates is that there are purple and there are clear or yellow wells. And so in the purple wells, we can see that the tissues are viable, and where there's no color, that's less viability or no viability. So where we mark those tissues as dead. And what we see is we run those colors through a spectrophotometer and gain results here, where we will say 50% is our cutoff line, and this comes from OECD. and that 50% cutoff. And so if the tissue is above 50% viability compared to the negative control, then it's considered passing, so not an irritant. And if it's below that, we would say it's an irritant. So this is a great method that has had a lot of traction and discussion. So I think it serves us well to just highlight what we know about regulatory acceptance of this. So you can see the countries listed here where in those regulatory bodies where we're getting acceptance. I think the main point that we're most focused on as an industry is to gain acceptance with the FDA. They are a powerhouse when it comes to acceptance, and they can persuade a lot of other countries to accept or not accept the method by their example and their rationale. So I know we'll be talking about that later. I know Kelly will want to have a few words about where we're at with that. He's been a great advocate to have lots of discussions with the FDA. So I'm going to move on to just one more resource I wanted to point out, that all of us that have worked on developing that in vitro method wrote several papers. We did strive for most of them to be open source. Here's a list of the 6 papers we've focused on. Only one of them is not open source, but you can gain a lot of insight and information into the work that went into developing this method to understand that it's good science and is a great test to replace our current rabbit study that we have now. All right, I want to step into sensitization. This is the next test that's coming down the pipeline, pretty hot right now. And this is a complex systemic reaction. I put up these 2 slides. This comes from OECD. They did a document to talk about the pathway, the adverse outcome pathway that we study to understand the sensitization response. So this is quite a bit different than irritation, where you have an exposure, you remove the elements like maybe the detergent that you're adverse to, and the response goes away. But we know with sensitization that each time you're newly exposed, the response can be more intense. And so it's more complex for us to assess this in vitro. So I wanted to throw up this adverse outcome pathway, and I mentioned it came from OECD. I've referenced the document here if you'd like to pull that. That's available online. But there are several key events that we look for and have studied as an industry to identify methods we can use to capture these key events and understand the trigger pathways that will then happen afterwards. And so I bring this up because this is really the foundation of the work that's been done for finding in vitro or alternative sensitization responses. So if we look at ISO 10993, Part 10, which covers sensitization, Annex C has an abundance of information of the work that's being done. And there's really 3 categories of assessments or types of assessments we're looking for, and that's in chemico, in silico, and in vitro. So I just want to give a highlight overview of what we're looking at for some of these as an industry. I'm not going to spend a lot of time on the in chemico because I wanted to give more time to the discussion, but again, there's great information in the standards. I wanted to talk quickly about using some of the chemistry that I know has been used around the globe for other industries. The main hangup I'm finding and that I hear about is that there's not appropriate thresholds or widely accepted thresholds for sensitization. So what that means is if you run the chemistry and you have these unknown values, which happens as we run our screening, we have to be able to say if what could possibly be in those unknown values is either below or above an acceptable threshold for the endpoint we're evaluating. So if we're looking at sensitization, we need to know if we have, 1.5 micrograms of unknown can we say, even if that's a sensitizer, it's not going to elicit a response? We're confident of it. So I just wanted to highlight some publications that are out there about applying some of these thresholds in the industry. And I think we're having great discussion. And I'm sure Kelly and Ron will probably want to add to this as well. And this is one of the topics, if we can have a widely accepted threshold, there's more probability to move into the chemistry, so in chemico and in silico, because it will be some chemistry, and then also using some computer, literature information to assess the endpoint. And then I wanted to point out for the in vitro methods, there are so many methods that are out there that are really promising for the industry. I didn't want to spend time going into each one of those specifically. There's definitely some that are getting a little bit more traction than others, but right now, everyone is looking for what method is most reproducible and will provide us the best results and can be performed in labs widely if we can have the expertise and the equipment to run those assays. So in 10993-10, there are these tables for each method. I just pulled this for census, just randomly here, because I really like these sections at the bottom, application to medical devices. It gives an update of where these methods are and how they're applying to medical devices. And some of them still need to address maybe one or two more things before we would readily apply them to medical devices. So I encourage you to read 10993-10 and go to the Annex C so that you can review these tables and educate yourself on these methods, because they will be getting some traction down the road. Kelly and I are on the ISO working group as we are partnering around the globe to begin some round-robin testing. That's the next round that will happen with sensitization. So this is the next test that will have wide traction in the medical device industry. And then I just briefly wanted to touch on 3 other tests that are starting to get some traction. And we have thrombogenicity, and the most common test done here is a dog thrombogenicity study. And there are an abundance of issues with this in vivo test, and it's very difficult with no standard acceptance, and it's difficult to get replicates because of the size of the animal and the number of animals that need to use. So there has for a few years now been discussion of a blood loop study. And this is where it's just what you imagine. You take a loop of tubing, you tape it together in a loop, we run it through a peristaltic pump, and to simulate pumping, circulating blood. We're able to insert a device, treat it very similar to an animal model. And this is gaining traction on acceptance for some medical devices. We've had success on this with the FDA. It really depends on the exposure time of the device with the actual patient, whether this model will be accepted by the FDA. But I'm really pleased that they're opening that conversation so we can use this method where it makes sense for short-term circulating blood contact devices. In the hemolysis realm, there are studies. Currently, hemolysis is commonly run using rabbit blood, but there are studies to show that we can get reproducible, similar results with human blood. So the goal would be to use human donors for the testing. And then with the pyrogenicity assay, there's the monocyte activation test. So this is a great test that actually can be used to monitor multiple types of pyrogens. So it can do bacterial endotoxins, and it can also look at material-mediated pyrogens. So it's nice because it combines a couple of different tests we're doing now in the industry. Right now, it is used in the European pharmacopoeia, so for pharmaceutical products, and we're having discussions to move that along in the medical device realm. So we're not there yet for med device totally, but again, this one will be moving in that direction soon as well. I've also just added an acronyms slide here because I believe a lot of these will come up in the discussion today. Some were mentioned in the presentation in the slides previous, so this is for reference after the fact. And then I also just, as I end the presentation and we move to the discussion, just want to highlight that Nelson Labs is forming an academy where you can go online and tutor yourself in several of these disciplines where it's concerning testing for medical devices, not just biocompatibility, of course, sterilization, hospital reprocessing, packaging, and just a wide variety of information. So we've taken our content and really made it very searchable. We're also going to create learning pathways so that you can train experts in your own facility and be more supported there with expertise. So that's where I'm leaving the presentation today, so we can move on to discussion and question with this panel. I think Thor, I'm not sure who's taking the reins, but I know it's not me. It might be Thor.
It's me. Thank you, Audrey, for all that presentation and for your willingness to kind of tee up Kelly and Ron and I. So thank you. I think the first question that I kind of wanted to discuss, and Ron, I think this is probably to you first, which is, right now the irritation, the in vitro irritation test is in the MDDT program. And maybe this is Kelly too, because I know, Kelly, you've been working on this with both of you. What is the biggest hurdle right now? I know that this in vitro irritation's been in the program for a very long time. So what's stopping us from moving this forward through that program? So Ron, maybe you first.
Yeah, so that's a great question, Thor, and it's certainly central to the main theme of the presentation about regulatory acceptance. So I think we could use that as a good case study. It might be worthwhile to just step back for a minute and just explore what the MDDT program is, and I know Audrey had mentioned it, but when I was a toxicologist at FDA, we would often have submitters come in and say, we would like to use this in vitro assay. And it's been validated by OECD or some other august body. And we would say, yeah, that's great, but how applicable really is it for the kind of compounds that we know are released from devices? Audrey had pointed out that Annex C that lists the applicability domain for compounds, for each assay, and how does that relate to the endpoint that you're trying to assess. So this medical device development tools program really offers a pathway to qualify your new method for regulatory use. So that's the term that FDA uses, to qualify it. And it really goes beyond just validating it. And it really shows that your assay is applicable for the endpoint that you're looking at in the device realm. So your conditions of use, your context of use. And so that's very important for FDA. Now, there are different kind of MDDTs that can be developed. Some are biomarkers, some are clinical assessment tools. But the ones that we're most interested in, the ones that Audrey talked about, are non-clinical assessment tools, either in vitro models or computational models. So the same types of principles apply. So just specifically, and Kelly, please jump in, rather than identify any specific roadblocks, it might be worthwhile to look just generally, what is FDA interested in seeing in an MDDT application? And I think a lot of these are relevant to this specific application. So I mentioned applicability domain. They're very interested to see, can your new test, whether it's a computer model or an in vitro model, really pick up the kind of endpoints that are relevant in a biocompatibility assessment. So let's say you've got a test and it was developed using pesticides, and it works great to predict the toxicity of pesticides, but we don't necessarily know that that's going to work for the kind of compounds that are released from devices. So it's important to be very clear about that. And I know that that's a concern that FDA has about the in vitro irritation test, to make sure that we're able to demonstrate that the test really works for the kind of compounds that are released from devices. Kelly can get into a little bit more detail, but it's really a two-pronged approach. First is demonstrating that the test method itself works for these kind of compounds. And then to show that the kind of compounds that it was validated against look like and act like the ones that we know are released from devices. I think the other thing important too is when people discuss applicability domain, a lot of the times they just talk about the chemistry. But I think it's equally important to talk about the biology as well. So Audrey was talking about AOPs and the extent to which we need to take mechanistic considerations into account. So I think that's important as well. I think the other thing that FDA is concerned about in general, but in particular with this test, is the context of use. So again, let's say you've got a test and it works really great for aqueous extracts, and then you're trying to say, is this going to work for the irritation of any kind of compound, let's say it's in a gel, let's say it's high pH, low pH, what have you. Is it volatile? Different physical chemical characteristics. So those are two things that FDA really wants to see. They also want to see, does it work well with a range of potencies? Can it pick up irritation or whatever endpoint for weak irritants or weak sensitizers? And then how does it work under clinically relevant conditions when you've got a complex mixture going on? So I think those are four aspects of the MDDT program and qualifying a test that make it a little bit different than just validating it. So in my mind, validating it really is, does the assay work? Does it do what you intend it to do? And from a practical perspective, how easy is it feasible to do? Can other labs do it? Is it reproducible? Those kind of things. So the MDDT program really takes the next step, and is your assay applicable for devices?
So Kelly, on that, what are we? Oh, sorry, Ron, go ahead.
Oh, okay. I was just, no, please, Thor, go ahead. I was going to ask Kelly what his perceptions on that were, because we've been in a lot of the same discussions with.
Yeah, I was just wondering with Kelly, like what's stopping the in vitro now? I feel like Ron's points are valid, and I think they're legitimate concerns that the FDA should have. But I also feel like with the in vitro right now, we've tried to address a lot of those. So what do you see as being kind of the barriers right now for this in vitro test?
Well, I think we're approaching the finish line. Believe it or not, it's I think it was last June when FDA posted on their Recognized Consensus Standards website that they said they recognized the in vivo portions of Part 23, but not the in vitro portions. So I'm hoping that this June, maybe that will change. But we've had a series of discussions with them, and they had several. I think it was about 4 specific things that they were concerned about. Ron's hit on them, some of them already. Context of use was a big thing, and regarding that, I guess the simplest way you could put that is the tissues that we use, these 3D human tissues, they're designed to replace the rabbit test for most medical device uses, but they're not designed to replace, eye irritation testing or mucosal or penile or vaginal irritation testing. There's other assays, 3D human tissue assays and other types of ocular assays, non-animal assays, that someday I think will be validated for medical devices, and they will make their way into, Part 23 or maybe another, Part 24 or 25 ISO standards. So context of use is important. So we're focused on the main type of irritation, not those other additional types that are in the one of the annexes of Part 23. I can't remember which one it was, Annex D maybe. So context of use is important. Ron touched on the chemical domain space. That was one of the key issues with FDA. And I mean, if I was in their shoes, I'd probably ask the same question. I'd say, well, how do we know that these tissues, I mean, they were validated years ago for industrial chemicals, a wide variety of industrial chemicals. We used a much smaller subset of irritants in our medical device validation study. However, how can FDA or any regulatory body be confident that these tissues will work well with medical devices? And we have some great data from Ron. He's helped us with this. And one of the best or most obvious pieces of information that maybe we missed initially is there's a set of, they call them performance chemicals. And if Nelson Labs wanted to create its own tissue now, or call it the Thor tissue, we can use some of your skin, Thor, and grow up these tissues. If we wanted to do that, the Rollins series, get some from you and your brother. But if we wanted to do that, hey, we'll help you with it. But if you wanted to do that, you would have to validate these tissues and prove that they were comparable to the other tissues that are on the market. And there's a guidance document. It's called OECD 439. You can find it on Google really easily. It comes out maybe every year or so. And that's a list of all the, there's 6 or 7 of them now, RHE tissue models that have been recognized by OECD for testing chemicals. Not specifically for medical devices, but just for testing chemicals. and to get onto that list, because if you're on that list, that's like the Willy Wonka golden ticket, and you're guaranteed that people will use it because it's recognized by OECD and is considered to be similar or comparable in its accuracy and reproducibility to these other tissues that have been around for longer. However, to get onto the list, as I mentioned, there are these 20 performance chemicals, and they're a list that was, I think ECVAM published a list originally, and then OECD came out with a new list. I think the most recent list is from 2015, but with your the new Rollins tissues, you would have to correctly identify 15 out of the 20. And interestingly enough, 17 of those 20, we've been able to show Nelson Labs and other contract labs and my company, and I think Ron had a list from FDA and some other lists. 17 of those 20 performance chemicals have repeatedly been extracted from medical devices. The remaining 3, very similar chemical cousins of the remaining 3, have been extracted numerous times from medical devices. So that's a good starting point for our argument. We're talking about tissues that are validated with a set of 20 chemicals that essentially come from medical devices. Ron did some really nice work using the ICE database from NICEDM, and he was able to show that these medical device chemicals fall into the 20 performance chemicals, for example, fall into a much bigger chemical domain space of over 1,000 different chemicals that had been seen and extracted from medical devices that he had seen when he worked at FDA. So I think that particular question, I think, is pretty well explained. If we think about these performance chemicals, we think about this database that Ron did. It's what they call principal component analysis, where it's like Venn diagrams with dots, essentially, where you're showing that these chemicals are all you can look at many different physical chemical properties, but there's not that much difference from industrial chemicals to these medical device chemicals. So that was chemical domain, context of use, chemical domain space. What else? Mild irritants. We just had another paper that came out here, Christian Television, and a few other folks, that was published in Toxicology in Vitro. That was really nicely done and studied mild irritants with these RHE tissues, and also devices that are they call them non-extractables, which are not truly obvious to a lot of people. But I think ultrasound gel is probably the best one. It's not a drug. It's a device because it's used in a medical procedure, but it doesn't enter your body. But you can't, do extractions on it, or you destroy the gel. And the beauty of those tissues is you can just apply, gels or lotions, things like that, directly to the tissues, and they'll work fine. And the other interesting thing is rabbit test in general isn't that good at identifying mild irritants. It's maybe, there's a big study from NICEDM that was published last year. I think it's in regulatory toxicology and pharmacology. If anybody wants to know, just send us an email and we'll send you a link to the papers. And any of those other papers, we can send you links to them, also the ones that Audrey showed. But that paper was very interesting because they did a highly curated data study where they looked at a couple thousand different animal tests, and these are just rabbit irritation tests, with white New Zealand rabbits, and they wanted to see the same chemicals tested multiple times in rabbits in high quality papers in good journals with high Clemish scores. And they looked at the four, it was for EPA and also the EPA classification system, which I don't really understand very well, but the GHS classification also, which is what we would be more concerned with in the device industry. And they found that for corrosives, which are GHS category 1, and then for non-category, on the other end, those are non-irritants, that repeating rabbit tests over and over again, 80 to 90% of the time, you get the same answer. However, if you move down if those are the two ends of the football field, if you move towards the 50-yard line on one side of it, from corrosives down to I'd call them strong irritants. We used to call them R38 irritants. They're called category 2 irritants by GHS. There, it's about 60% of the time is the reproducibility rate. And if you move on to the other side of the 50-yard line, towards the non-irritant end of the field, is the mild irritant category. Those are called GHS category 3. Reproducibility rate there is in the 40% range. So not so good reproducibility-wise, the rabbit test, and its accuracy, it tends to overpredict irritation also. And these RHE tissues, the thing that's beautiful about them that Audrey mentioned is they're very quantifiable, and they're also very reproducible. So that gets us through three of these things: context of use, chemical domain space, mild irritants. Last thing I would touch on here regarding, and these are all things that FDA has talked about, and we've showed them slides and discussed this quite a bit, and have some more follow-up work to do with them. But the last thing is another fundamental question. They admit that these tissues do identify irritants that are, chemicals that are topical irritants, just like in, a rabbit topical irritation test or a human patch test. And but their question was, how do we know that these tissues can identify irritants in a similar way? Are they comparable to intracutaneous testing in rabbits? And there's two papers published now. One is the Nomura paper, and the other one is the Wim de Young paper. That's another follow-up paper to the big round rabbit study, where side-by-side RHE tissue testing and then intracutaneous rabbit testing was done with the same chemicals and got the same answers. So I think these four issues that I've talked about here in the last couple of minutes, we've got pretty good information on those. It's all written up. Believe me, Ron and others have helped, Christian Pelavision and Elena Kandarova, formerly from MacTech, and Wim de Young, formerly from RIVM in the Netherlands. They've all helped to prepare these written responses. And we're, I think the latest from FDA that, from my recall, I have to get back to this, they had a new data template that they want us to organize all our RHE information into a template to make it easier for them to look at in one place. So that's one of my missions here in the next few weeks, get this done in June, the 1-year anniversary of their non-recognition of our 23 standard. But we're going to provide that to them. And the other thing I wanted to mention is we thought, since there's other countries around the world that are sitting on the fence with regard to recognizing this, we've got all of Europe and most of Asia, at least the big countries in Asia. I think it would be really helpful if we take all this effort that we've put in over the last 6 months or so, writing up these very specific responses to these issues of FDA, because I suspect that these other countries are thinking the same thing. Great minds think alike. So they've probably got the same questions in their head about this assay. So we're going to take those, our written responses, which are, I don't know what they are, 60 or 70 pages, and boil that down, distill that down to something that's manageable, and get it published in a good journal. So then, and run up the money so it can be open access. And then everybody on Earth can have a look at that. See what we had to say to FDA and see if they agree with that. And if so, great. If not, then they can come back at us with some more questions. So long-winded answer. I got to get a drink of water.
Thanks, Kelly. And I can tell you that I don't think Rollins tissue is a very good idea. I've been told Rollinses can be very irritating by themselves. So we may not want to have the irritation response recorded with our tissues. So
we'll find some other volunteers.
Yeah, there you go. So another quick question for discussion, and then maybe we'll turn some time over to the audience since we're getting a little bit closer, depending how much we talk. But now that we've kind of addressed the irritation, what about the other of the most common tests, the sensitization? Ron, where do you see the acceptance of the sensitization? Do you think this has greased the wheels of regulatory bodies? Do you actually think that the sensitization and the irritation will be accepted regulatory in the next 5 years? Or do you think it's going to be another uphill battle?
So I can tell you that there are MDDT applications in the pipeline for sensitization. I don't think I can get into too many specifics, and the efforts aren't quite as far along as they are for irritation, at least in the U.S. But there's some very exciting work that's being developed. And Audrey talked a little bit about SenSIS and some of the other assays. Oh, it's up on the slide right here, with guard skin, for example, and, some of these other assays that target specific key events. So there's really a lot of activity going on here. Not all of it's being done in the medical device space, but one of the exciting things, I think, in this field is we can leverage technologies that are being developed for other fields of toxicology. A lot of this work, the catalyst has really been phasing out animal use for cosmetic safety assessment. And can we leverage the techniques that are being used there and apply them for our needs? And then address those questions that Kelly was mentioning in terms of context of use and applicability domain. So yeah, there's a lot of exciting work going on in the sensitization space as well.
Kelly, what do you think?
One thing I could add to that,
Yeah.
Thor, just for example, since Audrey's got this slide up here, US FDA now, or excuse me, US EPA is now accepting in vitro sensitization for their pesticide submissions. That's part of their whatever it is, the in vivo six-pack, because they're trying, EPA is on the forefront of a lot of this alternative testing with the federal government. And that's one of them that they've identified, and they believe that these assays are better than the animal tests.
So, Kelly, do you think that in the next 5 years we'll have in vitro sensitization and irritation methods for animals, or for biocompatibility, I should say?
Oh, sure. Yeah, I think it's funny. Years ago, when I first started working on this in 2009, back in the pre-Cambrian era, I thought, 3 years. It'll take 3 years. That's what we accepted worldwide, both of them. That's what I thought. Boy, was I wrong. One of our that fellow, his name is Daryl Underbecker. He was the vice president of research at Medtronic, and he's an animal lover. He used to oversee our labs, but very interested in alternative testing and reducing, the 3 R's and reducing the use of animals. And I made my pitch to him, and he was on some funding committee that gave us some grant money for pilot projects, which was really nice. And he told me, he said, I bet it'll take you at least 10 years to get that irritation alone approved. So the science won't be that hard. The rest of it's going to be hard. He must have told me that in 2010 or 2011. So he was right, because he'd been around for quite a while. However, I think with sensitization, we have a lot of advantages right now, because in one sense, it's been a good thing that irritation took so long, because so many interesting, in vitro sensitization assays have come online in the last 4 or 5 years. And many of them that are shown here on this slide, they're, except for the last 3, they're OECD 442 recognized assays. And some of the other ones, GARD and Census, may become OECD recognized standards in the near future. And if that happens, that would be great, because if we can use those, that's like the good housekeeping seal of approval. If you've got these assays that, they went through ECVAM validation and OECD certification and recognition, that really helps, I think, reduce the fear level, with regulators around the world, because they recognize ECVAM, they recognize OECD, they understand that these things work. And we've got this infrastructure or ecosystem of contract labs like yours, and government laboratories, and regulators around the world, and medical device companies, obviously, around the world, that are very interested in sensitization. And what was the number you told us once upon a time, Laura? How many sensitization tests has Nelson done, and what was your failure rate?
So I can tell you the failure rate is 0, right? So in my 20 years of running sensitization tests, we have never seen a failed sensitization test. We may have seen one that ended up not being a true sensitizer. But yeah, I have the number somewhere, but it's almost scary to say. So I'll have to go back to that, but it's a ton.
I thought you said it was thousands.
Oh yeah, easy. We're somewhere around the 500,000 guinea pigs that I've been through in my career.
Oh my God.
Yeah. So it's something that's very passionate for us, right? Because doing that many testing and not seeing a response makes you kind of wonder the usefulness of doing it. Either the manufacturers do so good at keeping sensitizers off the device, which I think is true, or the test itself isn't sensitive to pick up minor sensitization.
Exactly. Well, the other brief 2 things I could say briefly is, we have this ecosystem, like-minded people, they want this to happen. And now Christian Television has taken over as the convener of Working Group 8, because Wim DeYoung retired. And Christian had an interesting idea. At first, I wasn't sure exactly what he was talking about, but he wanted to make this in vitro sensitization cookbook, essentially. And that thing is going to be published pretty soon. Here's the cover sheet. I don't know if you can see it, but it's with a long name, and it's ISO. It's a technical specification 11796. It's got a really long title: Guidance for Interlaboratory Studies to Demonstrate the Applicability of Validated In Vitro Methods to Assess Skin Sensitization of Medical Devices. This is about 40 pages long. It's a nice document. It's been out for a lot of comments. Industry, I don't know what we submitted, 300 or 400 comments. FDA had a lot of comments. I'm sure you guys submitted a lot of them. And that thing is, as far as I know, I think it's going to be published this summer. And so that's a really nice outline, like a roadmap for how to do a in vitro sensitization validation study. It lists all these assays, and it's got a nice list of sensitizer chemicals that have been extracted from devices. Ron provided a lot of those, and other people helped flesh out that list. And it gives some really specific advice. It's really a method, manual on how to do this. And one other saving grace, I think, with this sensitization project is we're not going to try to make a whole bunch of polymers with, sensitizers in them, because that was the rate limiting step for the in vitro validation study, vitro irritation study. That took at least 3 years of trial and error. And you guys are intimately involved with that, Nelson was. And you felt our pain, you know how maddening that was. And so we're going to do, we're planning to do extraction studies on one or more types of medical device polymers, and then spike those polymers with some of these sensitizers, and then challenge, these different assays, and we'll see where it goes. But I'm optimistic, within 5 years and within maybe a year or two, irritation will be more widely accepted, and sensitization within 5.
Well, before we turn it over to audience questions, I want to see if Audrey thinks. Audrey, do you think that we'll have irritation and sensitization in vitro in 5 years, regulatory accepted?
I think we'll absolutely have irritation acceptance within that realm, hopefully sooner rather than later. I agree with Kelly's optimism on getting FDA acceptance, and it may be incremental, like for certain types of devices, maybe skin contact, or things like Kelly mentioned, gels that are very hard to test any other way, but this really speaks to that. They really fit into this method. For sensitization, I'm going to be optimistic about that. I think 5 years is a tight timeline. We need to get together as an industry and run these round-robin studies, which can take time because that's funding, coordination, agreements on a procedure, and getting that collaboration lined up, and then getting that data published for the regulatory agencies to review. So I think 5 years is optimistic for sensitization, but we're definitely shooting to go as fast as we can with it.
Thanks. Well, Tom, I do have a couple more, but I think it's time we hear the audience what questions they may have, and stop talking among ourselves. So, Tom, is there any questions from the audience that we can address?
There are a lot of questions for this great panel, so it's kind of an important conversation. So the first question, GLP regs do not have any requirements for chemical characterization testing, i.e. EL testing are not typically conducted under GLP. How does this impact EL data used for toxicological risk assessments? Is the expectation that EL should be done under GLP, even if it's not required? And then there's they'd assume, but those are the two questions.
I'd love to hear some of your guys' thoughts. I'll answer really quickly since, go ahead.
We do have an abundance of questions, so I think if we can...
Okay, so I'll just be really quick. GLP, since we do tons of GLP testing here, and we do ENL, which is not GLP, I can tell you that the GLP testing that we do has some very specific regulatory requirements, but the amount of quality built into the chemistry that we follow, that the requirements that they have in their instrumentation is much better than some of the GLP stuff that we follow with the animal testing. So right now, the FDA does not require the GLP to be done in chemistry, as long as the methods are still followed there within the quality system, and also that the methods themselves follow the chemistry specific quality system. So we're fine running it under the chemistry guidelines as it's stated without running a GLP.
Is there any counterpoint to that? Anyone want to argue with Thor? Take him down?
Audrey?
Not Thor, but not disagree, but just to support what he said. That first, Audrey, thank you for starting your presentation with chemical characterization and risk assessment. I think a lot of times we forget that, it's a relatively new technique, and it, one of the outcomes can be reducing animal use. So I appreciated seeing that. But yeah, Thor, you're right. We just don't have GLP. And, there are very active discussions about the best way to do that chemical characterization many of those revolve around quality. So I think that's already baked in to a lot of the techniques that are being used.
Great. Next question. What is the time frame of exposure to blood that you are seeing FDA acceptance for? Have we covered that?
No, we haven't covered it. I don't know. Go ahead, Audrey.
I think it's on the blood loop study, because I did mention that the FDA is accepting that. So we've had success with customers who have products that are less than 4-hour exposure to the patient. So if it's a quick procedure, 30 minutes, the blood loop study has been accepted in submissions with the FDA. So I would say the answer to that is a less than 4-hour clinical use. At worst case, it has to be worst case less than 4 hours.
Okay. We'll just move. Next question. Could you comment on the in vitro, in vivo correlation for the irritation and sensitization alternative tests? Anyone?
Kelly, that's right up your alley.
Kelly.
Well, we know
Center square for the block. Kelly Coleman.
Yeah, thank you. Yeah. Well, that's something that's been debated, I guess, for a long time. But rabbit test has been around since 1944, 1945. John Henry Draves published that paper. It was never validated for medical devices. I'm not sure if it was validated for industrial chemicals, but it is what it is. And it was taken off the shelf when the tripartite agreement was written in the late 1980s and then the early 1990s when the ISO standards, 10993 standards were coming out. I'm not sure, Ron, do you remember what year the Part 10 was first published? I'm drawing a blank on that. Yeah. Well, anyway, that's the skin irritation sensitization standard, the old one, and that was immediately adapted because that's the irritation test. And the problem with that test, from my perspective, is it tends to have a pretty high false positive rate because rabbit skin is much thinner than human skin. Epidermis is much thinner, and most humans, like me, we might have 30 or 40 hairs per square inch, but rabbits have 1,200 to 1,500 hairs per square inch. So rabbit skin is very porous. So it's thin and it's porous because it has so many holes in it. So if you put a chemical on the surface of it or just below the surface, doing an intracutaneous injection, for example, there are many drain pipes that could carry that chemical down to the metabolically active layers beneath it, which is why the rabbit test tends to overpredict irritation. I already mentioned the reproducibility problems with it compared to the RHE tissues. And the thing that's really neat about this protocol that we have, medical device protocol for the RHE tissues, the human epidermal tissues, reconstructed human epidermal tissues, is that we use a very long exposure time, 18 to 24 hours compared to industrial chemicals when they were exposed for, I think, 60 minutes. So if you've got an extract of a medical device, whether it's saline or sesame oil, if it's got any kind of irritant in it, these tissues are going to detect it without a doubt. So they have high accuracy and high reproducibility, much better than the rabbit test does. For sensitization, guinea pig test depends on what data you look at, 60 to 70% accurate. The device industry typically doesn't use the LLNA, and that's got maybe a 70 to 80% accuracy rate, but it's not used. These assays, especially the genomic ones, well, even the OECD 442 assays, as far as I know, with the 442 assays, you have to pass 2 out of 3 of them. Those are some of the assays at the beginning of the list that Audrey showed. Those have 80, 90% accuracy, and the genomic ones like GARD and Census, there might be 1 or 2 others. Those also have very high accuracy rates compared to humans for sensitization. So I think in vitro irritation, more accurate, more reproducible, and same story with these in vitro sensitization assays.
Yeah, so I think that's a key point when we're talking about in vitro to in vivo extrapolation. We need to take into account what in vivo data are we talking about. Mm-hmm. So we had a poster at SOT this past year with our friends from Sensigen with the GARD assay showing that, it didn't do a great job of predicting guinea pig maximization results because the guinea pig maximization test isn't that good of a test. But it does a very good job, as Kelly was mentioning, of predicting human response. So when we're talking about in vitro to in vivo extrapolation, we have to define which in vivo test system are we talking? What species are we talking about? So,
exactly.
Terrific. So I just want to say we do have an abundance of questions. We'll try to get to them all. If we don't, I'm sure our friends at Nelson Lab will follow up separately after the presentation. But so if you have a question, submit it, and they'll get back to you if we don't get it to it in the presentation. So next question, for a new product launch, do you recommend to run both irritation testing in order to establish trust with the in vitro method? Panel.
So I'll answer this one really quickly, and then I'd love to hear your thoughts. That's one of the questions I was going to ask Ron and Kelly, is what can people do to help us, right? And we do have some partners. We published data from Arthrex, for example, where they ran a bunch of in vivo and in vitro comparisons side by side. And that really helped us show the FDA that in real life we're able to compare. So for me, if people are willing to help, or if you want to show confidence in the in vitro, it's something that we should look at is running, and I'm never going to try to sell more testing, even though I'm from a CRO. It's against my policy. But in this particular case, especially since it's not in vivo testing, it is beneficial for us to start having some real life data to compare in vitro to in vivo on the same products from the same lot. So, but from a regulatory standpoint, a lot of the regulatory bodies are already accepting the in vitro irritation. So we're not really needing those comparisons except for kind of help prove concept to like FDA, for example. I know Kelly, Ron, do you guys have other thoughts there?
I know, at least in my company internally, there is, I can't even tell you, maybe a third of the irritation testing going on is dual testing. And then some of it is just in vitro irritation testing, on products that have been on the market, and so, like EU MDR follow-up, gap analysis stuff from products that were marketed, 10 or 15 years ago. So now they're going back and they're looking at what was done back then, what kind of chemistry and biotesting was done, and they're oftentimes just running in vitro irritation again. So we're getting dual data sort of through the back door with those. But our toxicologists have been collecting that. We had a poster some folks put together at Medtronic showing at SOT, I think it was 15 or 20 products and dual data. It was triple data because they had cytotox data also on all those things. And internally, we've been debating, should we, talk to AdvaMed or Life Science Alley or one of these, industry groups and set up some little task force and encourage people if you're running into the same dilemma that we're running into, and you're doing dual testing, would you be willing to submit that data? It could be blinded data. We wouldn't have to know what company it came from, and just a generic explanation of what the device is to build some bigger database.
Great. So let's go to, we're going to try to get to at least one or two more questions. So first we'll go with going off Ron's condition of use issue. Would there be any mechanism of irritation that would not be detected with a measure of viability via MTT of the 3D skin model? So is that for you, Ron?
Yeah, and I'd welcome other people's thoughts as well. To be honest, I've really been focused in on mechanisms of skin sensitization and haven't given quite as much thought to mechanisms of irritation and whether or not the existing assays can cover a wide range of mechanisms. Kelly, maybe you've given that some thought, but maybe one way to address that in a general sense is if the test does a good job of being able to predict in vivo irritation for a wide range of compounds, we assume that wide range of compounds acts by a variety of mechanisms, not just one mechanism. So I think that suggests secondarily that it can cover multiple mechanisms. But any thoughts on that, Kelly?
Well, a good point. Is it disrupting cell membranes? Is it these irritants? Are they soldating them, or is it an ionic thing, or blocking transport channels or something? Irritation is different things, but it comes down to MTT. Obviously, it comes down to cell death. So one way or another, you're killing those cells. You can kill them a lot of different ways. As I mentioned earlier, we have very long exposure times with these tissues. So even if you've got, weak or mild irritants, they will be detected with these 18 to 24-hour exposure times.
Great. All right. We'll go into a bonus round. We'll go a couple of minutes over for question number 13. Approximation of turnaround time reduction for irritation in vitro versus irritation in vivo.
So this is an interesting question. I'll leave it to Audrey, but I want to say it's interesting right now because we're seeing such balloon turnaround times in the industry right now with the backlogs and the animal tests that that's one of the reasons why I feel like we should really be looking at acceptance in vitro because of the current state of biocompatibility testing especially in North America. And so we potentially could see a very good reduction in turnaround time, which means that these devices could be to the market quicker with now than ever before because of this. But Audrey, I'll let you do the answer.
Yeah, I just wanted to just outline for perspective. So an in vivo test typically is about 5 weeks. An in vitro test is also about 5 weeks. But like Thor said, with the delay with the vivariums getting the animals, we're seeing that 5 weeks extend up to 7 weeks, which we don't have the same delay with the in vitro tissues. One thing to consider too, once the in vitro method is widely accepted as a CRO, then we can have a standing order of tissues where we're not waiting 3 weeks every time we get a study in. We can have a standing order of tissues knowing that we will fill that plate of tissues with samples so it can cut the testing down to about 2 weeks. And if we can incorporate sensitization into also using those same tissues, that test goes from 8 to 9 weeks, which right now is 12 weeks with delays, to less than a month. I mean, and these tests are limiting factor tests in getting you to a market. So it could be really beneficial from a time perspective.
Another reason why people should try to push their own agendas for this, right? To try to get acceptance.
It will help everyone.
Excellent. All right. Well, I think that's a strong question to end on. We do have some more in the queue. As I said, Nelson Labs will get back to you and we'll respond. Thank you for the many questions. It's always a sign of a great and informative presentation when more thoughts are generated. So clearly a good job by our panel today. Thank you very much, Audrey, for your presentation, and Thor, Ron, and Kelly for your thoughts that followed.
Sure. My pleasure. Thanks a lot, everybody.
Thank you.
And
Thank you.
And thanks, of course, to everyone who tuned in today. It's great to be back on Device Talks Tuesdays. We'll be back again next week, so make sure you register and return. Have a great night or afternoon or whatever you got coming.